Now we'll welcome to the stage ISMB 2018 Chair Christina Rango to introduce this morning's keynote, an ISCB Innovation Award recipient.
It's a great pleasure to introduce Madan Babu, who's going to be awarded the Innovator Award. I've known Madan since 2000, I think. He came visiting UCL. He was looking for a PhD somewhere, so he was visiting several colleges in the UK. And I remember being very impressed and thinking he was an exceptional student. And anyone who managed to attract him to their lab was going to be a very lucky group leader. He'd come from India, from the Indian Institute of Science in Chennai, where he did a degree in biotechnology. And he made the very sensible decision of going to study with Sarah Teichmann for his PhD, where he looked at the structure, evolution, and dynamics of transcription factor regulatory networks. And he did, I think, some very outstanding work then, really completing the transcriptional landscape of mammalian genomes. Following that, he had a one-year fellowship in the States where he went to work with Aravind, and he looked more at the evolution of transcription factor networks. And then just after that very short visit, he returned to the LMB in Cambridge, where I think he became the youngest group leader, tenured group leader at the LMB. And he was in the unit of regulatory genomics and systems biology. Just 4 years later, he became the program leader of that unit. And it's really been wonderful, I think, to watch the progress, and have not been surprised either by the very impressive scientific trajectory and by the groundbreaking work that he's been doing. He uses structural biology, genomics, machine learning to study very important classes of proteins. In particular, he's looked at the GPCRs and at intrinsically disordered proteins. And as you know, GPCRs are very important therapeutic targets. 30% of drug sales relate to these proteins. And Madan has done some very elegant characterization of variants in human proteins that lead to disease. And I think this work will clearly help to find more specific drug targets for these proteins and obviously aid human health. He's also made very fundamental discoveries in intrinsically disordered proteins. Up to 40% of human proteins have these regions. And he's really helped to discover how these regions alter the functional versatility of proteins by changing the localization of these proteins, and explain how these regions contribute to cellular regulation. More generally, he's interested in studying the interplay between regulation and genome evolution. And his impressive work has led to recognition by many societies. There are too many awards for me to go through them all, so I'm just going to highlight the awards that he has from the last 5 years. As well as the Innovator Award that he'll receive today, he's been awarded with the Blavatnik Award, which is the UK Life Science Laureate. Last year, the Chemical Society acknowledged his work by making him a Fellow of that Royal Society. He was became an elected member of EMBO in 2016. In 2015, the Royal Society acknowledged his work with the Francis Crick Medal. And in 2014, the Protein Science Society made him a Young Investigator. In 2014, he got the Lister Institute Research Prize, and the Biochemical Society acknowledged his work in 2013 with the Colworth Medal. So just a selection of his awards. And I want to end by saying, as well as this very impressive work, I just want to say on a personal level, I think he's one of the kindest and most generous colleagues that I know, and I'm really looking forward to hearing about his work. So please welcome Madan Babu.
Thank you very much for your very kind and generous introduction, Christine. And good morning, everybody. So first of all, I must say that I'm really delighted to be here, and I'd like to thank the person who nominated me for this prize, the ISCB Awards Committee, as well as the organizers for giving me this opportunity to present our work today. But I thought I should first start off by giving a very brief personal anecdote about how ISCB had an important impact in my scientific career. So the first ever fellowship that I received was when I was a first-year PhD student, which was the ISCB Travel Award from the ISCB to attend RECOM and present our work as a form of a poster. And this was one of the most important events that happened very early on in my scientific career. And this really allowed me to meet a lot of my scientific heroes and mentors at the time. And this is a picture of the website from 2003. And we were some of these travel awardees who were featured on this website. And for some of you who might know, so that's Dana Peer, who also received the travel award at roughly the same time. So I have a really great affiliation to ISCB, and I feel very grateful and honored that I'll be receiving this ISCB Innovator Award this year. And I must also say that I'd like to thank the organizers and congratulate the organizers for such an outstanding set of talks and scientific topics that are covered in ISMB. All right, so what I'd like to do in the next 40 minutes or so is talk to you about unstructured proteins and how they actually contribute to increasing cellular complexity and also contribute to human diseases. But before I do that, what I'd like to do is to spend a slide giving a brief overview of the kind of problems that we work in my group. So our group is interested in understanding how regulation is achieved in cellular systems at distinct levels of complexity: the molecular level, the systems level, and the genome level. So at the molecular level, we are interested in understanding the structure and function of uncharacterized proteins and important regulatory molecules such as G protein-coupled receptors and transcription factors. Now at the systems level, we are interested in understanding how the different regulatory processes influence each other in order to maintain cellular homeostasis and how the breakdown of this gives rise to disease outcomes. And finally, at the genome level, we are interested in understanding the interplay between regulation and genome evolution. So how the requirement to achieve all these regulatory properties constrains the way in which genes and genomes evolve, all with the ultimate goal of being able to understand the molecular basis of diseases caused by regulatory dysfunctions at different levels of complexity. So as Christine mentioned, initially I was thinking about two different topics that I thought I could present. So one focused on G protein-coupled receptors, where we had looked at the structure, function, evolution, and pharmacogenomic aspects of GPCRs. And this involved systematic integration of multiple large-scale data sets that are quite disparate and diverse, ranging from drug information to genetic variation data, structural information, functional effects, and sales information. And each one of these aspects would potentially be of possible interest to some of the communities of special interest that I've highlighted here. So if anyone is interested in discussing more about this aspect of our work, I'll be more than happy to talk to you about this work after this presentation. But what I'd like to do in today's talk is to first give a very general introduction to what exactly are intrinsically disordered proteins. And I'll then highlight by discussing some of our earlier work on how disordered proteins contribute to cellular complexity and diseases. And in the third part of my talk, I'll be speaking about some ongoing work where we have developed some approaches that allows us to discover which part of unstructured regions are actually contributing to function. So this leads to high-throughput discovery of which regions are functional and what exactly makes some sequences more functional than the others. And finally, in the last 5 or so minutes, I'll have a very brief discussion about how intrinsically disordered regions could contribute to increasing the organismic complexity in general. All right, so as we all know, classic work from Christian Anfinsen and colleagues from the 1960s and 70s clearly highlighted that the function of a protein is encoded in the amino acid sequence, and this is achieved by the sequence of amino acids adopting a unique three-dimensional structure. And this led to Christian Anfinsen to postulate what is well known as the Thermodynamic Hypothesis, which highlights that the amino acid sequence determines the unique three-dimensional structure of the protein. And advances in crystallography and other structural biology methods and techniques, such as NMR and more recently electron microscopy, have really established what is called as the structure-function paradigm. So if we ask this question, how is function achieved by a sequence of amino acids, the classical view is that the sequence adopts a defined three-dimensional structure, which can have dynamics, of course, and the structure is important for function. And this is exemplified by the availability of numerous structures of enzymes, which highlight the importance of positioning specific chemical groups of amino acid side chain in spatial proximity in order to be able to catalyze enzyme function. But in the last couple of decades or so, there is an expansion of the structure function paradigm to address this question, how function is achieved by protein sequence. And this is what is referred to as the disorder function paradigm, where a sequence of amino acid can adopt an ensemble of different conformations. And each one of these different conformations can be recognized by other proteins in the cell. And in this manner, the disordered nature of this protein allows for the functional versatility by permitting these unstructured regions to interact with diverse interaction partners. And this is what is referred to as the disorder function paradigm. Now, importantly, several computational and some experimental estimates have also shown that over 40% of any protein that is encoded in eukaryotic genomes end up having these large disordered regions that are intrinsically unstructured. So what is the formal definition of disordered protein regions? So I'll be referring to intrinsically disordered regions as IDRs. So IDRs are polypeptide segments that lack a unique three-dimensional structure, either entirely or in parts. And it is also believed that they sample a variety of different conformational states that are in equilibrium within each other under normal physiological conditions. And therefore, if you think about protein and protein conformations and structures, rather than thinking about them as just being consisting of structured regions, one could think about them as being in a structural continuum, where on one end of the spectrum you have proteins that are well behaved, that fold into defined three-dimensional structures, and these kinds of proteins have been studied in great detail, and very early work has really highlighted the importance of the kinds of protein functions that can be achieved by these protein domains. And then you can have the other end of the spectrum where you have proteins that are fully disordered, but then you also have those in between where they can have different levels and amounts of flexibility in them. So the first question that comes to mind is, why do these proteins not fold? Now, one of the early work which looked at the amount of charged residues and the amount of hydrophobic residues basically highlighted that most of the proteins that end up folding end up having sufficient hydrophobic residues that basically drive them to cooperatively fold into defined three-dimensional structures. Now, when you look at many of these unstructured regions, they do not have sufficient hydrophobic residues. They tend to be well solvated, and therefore they are much more happy to not fold and actually interact with the water molecules, and hence they end up having more intrinsic flexibility within them. But then there are also some unstructured proteins that fall at this cusp, which have some hydrophobic residues, but not sufficient to allow them to drive the formation of a defined tertiary structure. However, when the right interaction partner comes along and presents an appropriate chemical surface, these proteins tend to adopt a fold when they interact with this protein. And this phenomena is what is referred to as coupled folding and binding, where the protein can adopt a defined shape when they interact with the right interaction partner. Now, very important in early work from Cristina Ringo and co-workers early on also ended up addressing what kind of functions do these intrinsically disordered proteins participate in. And their studies showed that some of the functions that they primarily participate in involves transcription, signaling, and also membrane-related processes such as membrane proteins. Now, if they don't adopt a defined structure, the question comes to mind as to how exactly then do they achieve their function. Now, one mechanism by which they contribute to increased functionality is through their conformational flexibility. As I mentioned, many of these proteins adopt different conformations, and different conformations are recognized by different proteins. So here is an example of what's called the HIF1 alpha protein. So the HIF1 alpha protein interacts with a transcriptional coactivator called CBP, and when it interacts, this red region, which is typically unstructured, ends up adopting an alpha helical conformation. And there is a critical asparagine here that mediates the interaction with the CBP protein. However, the same protein can be post-translationally modified at this critical asparagine by this enzyme called asparagine hydroxylase. And in this case, the protein becomes completely unstructured, and the asparagine is presented very deep into the active site cleft. So it's this conformational flexibility that allows different proteins to now interact and come up with very interesting dynamics and kinetic effects that allow protein-protein interaction regulation in a very serious manner. Now, the other mechanism by which disordered regions contribute to function is by providing a very flexible environment to be able to present what are called as short linear peptide motifs. So these are short stretches of amino acid sequences. An example is shown here, which is called as a Ken box motif, which is recognized by one of the E3 ligases, CDC20. And this recognition is mediated by these short peptide motifs. And if you can imagine a large disordered region, you could once very quickly also identify the possibility that you have many different short linear motifs in a single disordered region, and this can allow assembly and disassembly of multi-protein complexes dynamically in space and in time. So it provides this enormous flexibility to be able to mediate simultaneous interactions with many different proteins in the cell to coordinate and regulate cellular processes. And in this manner, many of these intrinsically disordered regions also emerge as global hubs in protein interaction networks. The other major mechanism by which disordered regions contribute to function is via post-translational modifications. Because every chemically modified entity is a distinct chemical component in the cell, the same protein can therefore exist in different chemical states depending on which residues get post-translationally modified. And a classic example is highlighted in the histone tails, where the proteins can be modified by methylation or acetylation or ubiquitination and phosphorylation and so on. And different modifications and combinations thereof are recognized uniquely by different protein interaction domains that then allows them to assemble in space and time, and therefore also permits the same protein to participate in multiple different cellular processes. For instance, histone tail, through their modification code, can contribute to many different processes ranging from transcription to DNA repair to replication. And even within a particular type of process like transcription, depending on the nature of the modification, it could lead to activation of gene expression or repression of gene expression. And all of this is possible through these modifications. And this, therefore, serves as a very good substrate to encode and decode information through individual post-translation modifications and their combinations thereof. Now, another very interesting aspect of intrinsically disordered proteins, which is something that has been noticed by several scientists early on, including Mark Gerstein, is that these intrinsically disordered regions tend to evolve very rapidly compared to protein structure domains. Because they do not have the requirement to adopt a defined three-dimensional structure, they're much more tolerant to certain kinds of mutations, and therefore these regions tend to evolve much more rapidly in the protein sequence space. For example, if we start off with a random sequence, within one or two different mutations, we could very quickly evolve a linear motif that can mediate a domain peptide interaction. Or you could also evolve a residue that can be post-translationally modified, and hence you can regulate these interactions very, very quickly. So this property of these intrinsically disordered regions basically allows motifs and modification sites to emerge and disappear very, very quickly in an evolutionary timescale, and therefore new interactions can be made or broken very quickly within a short evolutionary time span. Now, this is a property that has been exploited by viruses. Our group and several other groups have also shown that many of these viruses tend to evolve very rapidly, also encode disordered regions, and also end up acquiring, through molecular mimicry, certain motifs that mimic the host cellular motifs. So in other words, the viral proteins end up acquiring the same set of short amino acid stretches, the linear motifs that allows interaction with many of the host cellular processes in order to be able to mediate successful infection. And this rapid evolution of these motifs can therefore rewire host virus protein interaction networks to mediate successful interaction. Now, I highlighted many of the advantages of the unstructured regions. There are also potential dangers of having these intrinsically disordered regions. Depending on the nature of the amino acid substitution, these unstructured regions could also become aggregation prone, and therefore many of the mutations within disordered regions have been associated with a number of neurodegenerative disorders and diseases, such as Parkinson's, Alzheimer's, and so on. So here's an example where people have analyzed many of the plaques that are formed as deposits in many different types of neurodegenerative disorders, and these aggregates have been enriched in intrinsically disordered regions. Whether these are causal or whether these are consequence is an area of active debate, but it highlights that if you overexpress these proteins or certain kinds of mutations can lead to transient loss of function by acquiring them and storing them in these large deposits. Now, many of these intrinsically disordered regions, as I mentioned, are also featured in transcription factors and signaling molecules like kinases and membrane proteins and membrane protein receptors. And many of these also end up being proto-oncogenes and tumor suppressor genes, and many different mutations and altered expressions of disordered proteins have actually been linked to diverse diseases, including cancer, simply because overexpression or altered abundances can lead to promiscuous interactions, because many of these peptide motifs are very short and small, and given that there are large gene families that are expressed in any given genome or cellular type, overexpressing them can lead to promiscuous interaction with other proteins in the cell that they should typically not interact with, and this leads to rewiring of protein interaction networks. And this is something that I'll be talking about later today. So this, at one point when we started our group, raised a question. So if disordered regions are so versatile in the ability to interact, rewire protein networks, and are functional in many different contexts, and at the same time they can be potentially dangerous because they can form aggregates, or they can also kind of like rewire protein interaction networks inadvertently, giving rise to disease outcomes, how exactly do cells minimize the detrimental effect of disordered proteins, and at the same time allow them to permit all these different functions within the cell? Now, to address this question, in 2008, what we did was to systematically integrate and combine multiple large-scale data sets that describe different aspects of gene regulation, ranging from information about transcription rate, steady-state levels of transcripts, transcript half-life data, as well as information about protein translation rate, protein abundance data, and protein half-life data. And we compared all the proteins that are enriched to have a structured domain compared to the ones that had large disordered regions. And what we found was that at each and every stage of the central dogma during the process of gene expression, proteins or transcripts that encode disordered regions tend to be much, much more tightly regulated compared to structured domains. So if you think about the availability of the protein in terms of their abundance or the amount of time that a protein spends in the cell, as long as the proteins that is unstructured are present in low abundance and spend short periods of time, you have what is called as a desired outcome. And by desired outcome, the protein remains soluble and you mediate the cognate protein-protein interaction in cells. However, if you increase the abundance of these proteins, or if the amount of time that is spent by a protein in the cell is increased, then you go towards the undesired outcome. And by undesired outcome, I mean that the protein starts to form aggregates, or they may end up mediating signaling tone. So it is this coordinated and tight regulation of disordered proteins at different stages during gene expression process that ensures that these proteins are present for short periods of time and in low abundance, and this ensures that the detrimental effects of these proteins are minimized. And many of the mutations that result in disease affect either their abundance or their half-life, and these contribute to disease outcomes such as protein aggregation and signaling crosstalk. So what I'd like to conclude in the introduction is that intrinsically disordered proteins should not be viewed merely as passive linkers that bring together structured domains, but these can be viewed as fundamental units of protein function. And what I'd also like to highlight is that compared to structured domains, intrinsically disordered proteins are fundamentally different in many different aspects and attributes. For instance, the kind of amino acids that form the sequences are very distinct. The kinds of molecular forces that allow these proteins to sample different conformations are distinct. The kind of conformations that they could sample are also different. The kinds of function that they perform are different, and they also have very different evolutionary rates compared to structured domains. This also does not mean that all disordered regions belong to the same group and cannot be clubbed as one big entity. Just like how structured domains come in different flavors, like the all-alpha helices or the all beta proteins and alpha beta proteins, disordered proteins can also be grouped and classified into different types depending on the kind of functions that they perform and the kind of attributes that they have. Some have some hydrophobicity, which allows them to adopt a defined fold when the interaction partner is present. So these are called as coupled folding and binding. And others remain fully unstructured because they prefer to be much more well solvated. And some may present linear motifs, and others may present post-translation modification sites and combinations thereof. All right, so with this, I'd like to move on to the second part of my talk, where I'd like to highlight some of our work on how disordered proteins could increase the cellular complexity and also may contribute to different disease outcomes. So one of the problems that we were interested in understanding is to ask how exactly does tissue-specific splicing of certain disordered segments affect protein interaction networks. Now, this audience needs no introduction, but I'll just quickly highlight that alternative splicing is a molecular mechanism wherein certain exons are differentially included in some isoforms compared to the other, and which, when it is in the right frame and is not consequences of nonsense-mediated decay, end up resulting in inclusion of a protein segment in the final protein product. And this can lead to diversity at the protein level for the same gene that is encoded by an organism. Now, advances in next-gen sequencing technology have also highlighted that over 90% of the human genes have the potential to undergo alternative splicing, and nearly 50% of the isoform have tissue-specific distribution. There is also a lot of discussion and debate in the field as to whether many of these isoforms are actually made into proteins. That's going to be a topic of separate discussion, but for our study, we focused on instances where there was evidence at the protein level. So despite that, the roles of these unstructured segments are not fully understood or appreciated. So the question that we became interested in was to ask, given that a large fraction of the isoforms are estimated to be tissue-specific, what is the characteristic of these tissue-specific segments? Why does nature go through this trouble of kind of like exquisitely including or excluding these disordered regions? And how can the addition or removal of these segments influence the function of the protein? Now, to address this, at the time we looked at multiple RNA-seq data sets describing transcript isoforms from 10 different human tissues. And then we classified every exon into one of 3 types. So we have what are called as the constitutive exons. So these are the boxes. Every box represents an exon, shown in light gray. So these exons are always included in the mature transcript, irrespective of the tissue. And then we also have what are called as the alternative exons. So these are the dark gray boxes. The dark gray boxes are included in one isoform but not in the other, but they don't show any tissue-specific differences. And then we have what are called as the tissue-specific exon, shown in dark red boxes. So these are alternatively included in one isoform but only in one tissue but not in the other. Now, once we had this classification of different exon types, we then asked what kind of properties do these exons have at the protein level. So we find that tissue-specific protein segments, that is, those that are encoded by these tissue-specific exons, tend to be more often disordered compared to the alternative exons and the constitutive exons. And the constitutive exons or segments tend to be more often structured with very defined biochemical properties, such as DNA binding, or have enzymatic properties like kinase domains and so on. So if you think about a protein, the region that seems to be tissue-specifically included or excluded are not the structured domains, but these disordered tails that might be in the N-terminus or in the C-terminus of these proteins. Now, when we look at what kind of functions are likely to be affected due to the removal or addition of these unstructured tails, by mapping experimentally available biochemical evidences on protein-protein interactions and mass spec data on post-translation modifications, we find that many of the tissue-specific disordered segments that are included or excluded in a tissue-specific manner tend to be enriched in linear motifs and protein-protein interaction sites, as well as in post-translation modification sites. Now, to cut a long story short, we then mapped all this data onto available structures to systematically study what kind of impact do these tissue-specific splicing events have. So to address this question, how does tissue-specific disordered segment affect the function of the corresponding protein, we find that these tissue-specific splicing tends to affect unstructured regions rather than structured domains. That also has the propensity to contain interaction motifs and these post-translation modification sites. And what this immediately suggests is that this may result in protein-protein interactions and signaling events that occur only in some tissues and may hence contribute to tissue-specific phenotypes. In the next few slides, I'm going to highlight a few of these examples to give you a flavor of what kind of impact this mechanism could have in terms of larger networks and the kind of cellular processes and phenotypes that may result and emerge from this mechanism. So we found evidences where tissue-specific splicing tend to affect disordered regions with a linear motif that can create new interactions. So here is an example of a protein called phosphatidyl inositol phosphate 5 kinase. It's called the PIP5K gene. The isoform that is expressed in the brain has a short disordered region that exposes a peptide motif. And this peptide motif is recognized by a protein that mediates endocytosis, a protein that is called the AP2 protein. And this, therefore, allows the expression level to be regulated in the brain. However, the isoform that is expressed in the lymph node does not have this large disordered region, and therefore, even though the endocytic proteins are highly expressed, these proteins end up being constitutively expressed on the membrane compared to the brain version. So in network parlance, what this means is that even though the two proteins are expressed in two different cell types or tissue types, it mediates an interaction only in one tissue, but not in the other tissue. And this is because this is not because of removing a structured domain, but because of removing a short unstructured region that contains a linear motif. Now here's another general principle that we observe, which is that tissue-specific splicing tends to affect disordered regions with modification sites, and this can therefore create new substrates for signaling enzymes. And this involves a protein called Paxin-2. The isoform that is expressed in the breast tissue involves a disordered region that exposes sites that are post-translationally modified by casein kinase, and therefore this protein gets recruited as a substrate in the breast tissue. However, the isoform that is expressed in the brain does not include this region, and therefore even though casein kinase is expressed in more than these different cell types, in one cell type it recruits one protein as a substrate, whereas in the other cell type it is completely blind to the presence of this protein as a substrate. So again, in network parlance, you could rewire evolutionarily conserved signaling pathways in a tissue-specific manner and create signaling diversity and diversity in signaling pathways, not by affecting structured domains, but by removing short unstructured regions that can be post-translationally modified, or by mediating these domain peptide interactions. Now, I highlighted examples where it involved protein-protein interactions in a binary fashion, where there's one protein interacting with the other protein. We all know that many proteins end up forming large protein complexes, and this can also have a very interesting systems-level property and impact in terms of affecting the ability to form multi-protein complexes. And I'll highlight this using this toy example here. So here's an example where let's assume that you have two proteins that interact with each other to form a homodimer to form the active unit. If you end up expressing an isoform that has lost a part of this protein, you can now result in a situation where you can sequester this protein into an inactive complex. Now, if you also express multiple isoforms, this can lead to situations where it can compete for the endogenous protein factors. And this can lead to very interesting system-level properties and behavior, such as loss and gain of function, dominant negative response, and also what is called as an ultrasensitive response. I'm also happy to talk about these after my presentation. So I'll just highlight this using a real example here. So this involves the p53 protein. So in the case of p53, as we all know, it has multiple regions. The N-terminal region contains what is called as the activation domain. So this is an intrinsically unstructured region that is important for recruiting the transcriptional machinery to the DNA. Now, it also has a DNA binding domain that allows p53 to recognize specific sequences on the genome. It also has a tetramerization domain that allows p53 to come together and form a tetramer. So the protein forms a tetramer, binds a specific region on the genome, and through the unstructured activation domain is able to recruit the transcriptional machinery to initiate transcription. Now, there might be some times during development where the same set of genes that need to be activated needs to be shut down. And this is what happens in the case of p53 during embryonic stem cell differentiation. An isoform of p53 is expressed, where the DNA binding domain is retained, the tetramerization domain is retained, so which means the proteins can form tetramers. It can also recognize the same sequence on the DNA. However, the isoform lacks an activation domain. And because you do not have an activation domain, you have, in a sense, converted a transcriptional activator to a transcriptional repressor because it binds to the same sequences on the DNA, but is unable to recruit the transcriptional machinery. So in this manner, sequestration leads to the formation of non-functional tetramers that can still compete for the same DNA sequences. So what I'd like to conclude this part through this cartoon-like representation is that tissue-specific splicing of disordered regions can increase the functional versatility of proteins by rewiring protein-protein interaction networks through these linear peptide motifs or through altering post-translation modification sites. So you have two different tissues, where in one tissue you can include a particular exon or exclude a particular exon. This tends to be more often disordered, exposing a peptide motif for a post-translation modification site. Therefore, even though the same protein may be expressed in both these tissues, you mediate an interaction in one tissue, but not in the other tissue. So the differential inclusion of these disordered regions by alternative splicing thus rewires protein networks, and this changes the context in which the function is carried out by the same protein. So the kinase will still remain a kinase in both these cell types, but which protein gets recruited as a substrate can now be determined not by inclusion or removal of a structured domain, but because you can affect a disordered region that exposes these modification sites and linear peptide motif. So the way by which disordered regions therefore contributes to increased phenotypic complexity is by changing the context, but not affecting the biochemical activity of the structured domain. So this is just a cartoon representation to highlight, so you can use the same components, but you can rewire them differently in different tissues in order to create functional diversity. So there are a number of implications of these observations for evolution, for disease, genetics, and in general molecular biology and biophysics. My own personal favorite is for genetics because it allows intrinsically less evolvable proteins. So let's assume a developmentally important transcription factor, like a homeodomain transcription factor. You can substitute the homeodomains between organisms. The organisms are just fine. However, if you look at the tails of these homeodomain proteins, they evolve very rapidly, and they can actually mediate different interactions in an organism-specific manner. And this allows recruitment of completely different biochemical activities. It could be a histone acetylase or a deacetylase or methyltransferase to the same site, and through alternative splicing in a developmentally regulated manner, the same structure domain, which is the homeodomain, can now change different contexts by recruiting different proteins to the same site. So disordered proteins can be viewed as the yin and yang with structured domains in a multi-domain protein. One part is highly conserved, the other part is much more rapidly evolvable or regulatable, and that contributes to changing the context in which the biochemical function of a structured domain is actually presented to the cell. So a number of other groups have also observed what we have presented here, so which again highlights that this could be a general mechanism by which disordered proteins and disordered regions can contribute to increased phenotypic diversity in a tissue-specific manner by rewiring protein-protein interaction networks. All right, so what I'd now like to do is to focus on a different question, which is how exactly do gene fusions involving disordered segments affect protein networks in cancer? Now, as we all know, gene fusion is a molecular mechanism that happens at the DNA level, where two different parts of the DNA on the genomic or the chromosomes come together, where two parent proteins fuse together to create a fusion gene. And the fusion gene may result in a fusion transcript, and if they are in frame, and if you control for many other properties, they could give rise to a fusion protein. Now, fusion proteins have been characterized since 1980s. And since the early discovery of these fusion proteins as molecular drivers of cancer, there are more than 7,000 documented gene fusion events, but we still don't fully understand what is the actual effect of these fusion-mediated disruption of protein-protein interaction networks in diverse cancers. So the question that we became interested in was to ask, with so many different gene fusion events that have been implicated in diverse cancer types, what is unusual about these fusion products, and how exactly can these fusion products affect the known protein-protein interaction networks? And can we learn something about which proteins can emerge as parents for being able to form these fusion products that can give rise to disease outcomes? Now, to do this, we first generated multiple large-scale data sets of gene fusion events, thanks to Alfonso with his database, KITARS, which allowed us to systematically compile multiple gene fusion events that have been documented in the literature. And then for every fusion product that have been implicated in a particular tumor type, we then systematically looked at what the parent genes are, which part of the parent protein was included in the fusion product, and which part of the parent protein was excluded in the fusion product. And then we asked what is unusual about these proteins that contribute to generating these fusion products. And what we find is that the parents of the fusion proteins tend to be hubs in known protein interaction networks. And what this means is that any perturbation to these parent proteins are likely to affect a significant part of the protein-protein interaction network. And we also identified that parents of fusion proteins tend to contain large disordered regions that are also enriched to have linear motifs and post-translation modification sites. So parent proteins have high disorder content, high densities of linear motifs, and these modification sites. And what we also observe is that the fusion product preferentially joins proteins in the protein interaction network that previously did not talk to each other. So these are distinct parts of the protein interaction network, and the fusion protein basically acts like a short circuit, bringing together two distinct parts of the protein networks that are not normally seen next to each other. An example involves a protein that is highly phosphorylated, which is highly regulated through phosphorylation. Many of the fusion events ends up resulting in bringing a phosphatase right next to a protein that is heavily phosphorylated in a highly regulated cell cycle dependent manner. And this can result in a situation where constitutive dephosphorylation might lead to altered functions of these genes, caused primarily due to this fusion product. So fusion products that we observe in these data sets tend to permanently rewire and appear to disrupt critical signaling and regulatory networks. So I'm just going to highlight one specific example, which is representative of several other examples that we find in these fusion products, to highlight the molecular mechanism by which these disordered regions, linear motifs, and post-translation modification sites can lead to interesting outcomes. This involves a proto-oncogene called FLE1, which has a DNA binding domain, and it has an activation domain, as well as a site which is ubiquitinated. And ubiquitination allows proteins to be degraded in cells, and this is how this particular protein is regulated inside the cell. So this is a transcription factor. And then you have another gene which is called EWSR1, which is another oncogene that has a disordered region, and it also has ubiquitination sites. And gene fusion tends to avoid these ubiquitination sites. However, it brings a DNA binding domain in close proximity to a disordered region, and thus the fusion protein results in a transcription factor that can recognize certain sequences on the DNA and act as an activator because it can recognize through the activation domain the transcriptional machinery. However, because it has lost both its ubiquitination sites, this ends up becoming unregulated. So it's like a deregulated transcription factor that is highly expressed in the cell due to this fusion of these two distinct regions. So the loss of ubiquitination sites from an oncogene and a gain of disordered transactivation domain ends up resulting in a stable fusion product that acts as a strong transcriptional activator. And in this manner, fusion product in cancer appears to permanently escape cellular regulation. So it's bringing together different parts of the protein, loses its ability to be regulated by the cell. So it's like a rogue protein that has got a very strong drive or a function that can now no longer be regulated by cells. So this cartoon essentially highlights what I've just described. So gene fusion involves parent proteins, and let's say you have gene A, which through its disordered region mediates interactions and forms a particular protein-protein interaction network, and these tend to be hubs in these protein networks, and gene B, which also has a very similar phenomena. Gene fusion results in making these chimeric protein products that brings together two different parts of networks that don't typically see each other. And in this manner, they also remove sites that allows regulation of these protein products, and thus the escape of regulation and rewiring of disordered hub proteins in a permanent manner ends up altering the topology of these signaling and regulatory pathways, and thereby leading to potential disease outcomes such as cancer. At least that's the hypothesis. So in the Lego parlance for the Lego aficionados, so you have the same components, but this time you use Craggle. Okay, and then you end up permanently gluing some of these components, and therefore you give rise to potential disease outcomes. All right, in the next part, what I'd like to do is to present some ongoing and more recent work that we have been doing in our group, where we are trying to, in a high-throughput manner, try and identify and discover which parts of unstructured regions are actually functional, and which ones are merely serving the function of a linker and can therefore neutrally evolve during evolution. So this slide is again a summary to highlight that a large fraction of the proteins in the human proteome and any eukaryotic organism have large regions that are intrinsically disordered. They have many important functions such as transcription, signaling, as well as like protein degradation. They've also been implicated in a number of human diseases such as cancer and neurodegenerative disorders. And thus, there is really an enormous opportunity to truly understand a part of the proteome that has been completely ignored because many people have focused on structured domains. And therefore, there is also an opportunity to understand new biology that could be contributing to increased functional complexity of how proteins could actually perform such diverse functions. And I have not touched upon this point, but I'll be very happy to talk to anyone who's interested in. Many of these disordered proteins have unusual material properties, so they can actually end up forming hydrogels and phase-separated structures. And these are very, very interesting phenomena that have been described in the literature more recently, because you can form membraneless organelles. So even though proteins may be present in low abundance, by bringing them together in space and time, you increase the apparent concentration of these proteins in spatially restricted regions of the cell, and thus many of these regulatory proteins can have spatially localized execution of their function, even though they may be present in very low abundances, rather than have to overexpress them in very high quantities. So one of the key motivation in the field is that we now have a very broad general understanding about some of the general principles by which disordered regions can contribute to function, mechanisms by which they can actually contribute to phenotypic complexity, and so on. However, what the field was also lacking is basically to ask whether there is a targeted and a high throughput approach that one could use to discover in a simultaneous and high throughput manner which regions of the disordered segments are actually functional, And can we learn what is unusual about these sequences that allows them to be functional in a certain context? So this was one of the motivations for us to develop what is called as IDR screen. So where we try to use next-gen sequencing to study intrinsically disordered protein regions. So it is basically a discovery platform that tries to identify functional disordered regions in proteins and also tries to use machine learning algorithms to learn what makes some of these sequences functional. And this particular approach is a variant of many of what is called as an established approach. Many people have developed this, and we have basically tried to put them together in a systematic framework. And this is called as a deep mutational scanning in some contexts. And others basically use this as a screening strategy combined with next-gen sequencing. So we're just focusing on disordered regions, and that's why we thought it was important to label this slightly differently, to call intrinsically disordered region screening using high-throughput sequencing. So IDR screen is basically an approach where next-gen sequencing meets disordered protein regions. It has two major phases, an experimental phase that has three conceptual steps. The first step involves design of a selection system and validating it. So you basically ask whether a disordered region has a function of interest or doesn't have a function of interest. And then the next step is to use and create a library of sequences that codes for disordered regions. It could come from a genome of interest, or it could be a random library. And then you introduce this library into the selection system, and then you recover the sequences before and after selection, and then apply next-gen sequencing. So you know what is the complexity of the sequences that you have used for the assay. And after the selection, you know which sequences had the function of interest, and then the read depth gives you an indication about how strong the function is. And this is also perfect for machine learning applications because you don't only have true positives, but you also have true negatives as defined by this assay. And then in the computational phase, one could quantify which sequences survive the selection and then apply machine learning approaches to ask what exactly is unusual about these sequences that passed the survival screen and the ones that did not pass the survival screen, and therefore come up with general rules or define patterns that can explain functionality. So in this manner, what IDR screen allows is the discovery of functional regions within disordered segments of proteins, and also helps identify and define the rules that can make these disordered segments functional. So we first applied disordered IDR screen to study the problem of transactivation domains. So this is a problem that has been around in the field since 1980s. And very early work from Mark Tashney and others have basically led to defining what is called as acid blobs and negative noodles, which is like any stretch of highly negatively charged residues can actually act as a transactivation domain, but it turns out it is not that simple. So we thought this is a good opportunity to go and address this problem. So we focused on IDRs, and we particularly focused on transcription factors. And yesterday we heard this very excellent lecture from Martha Bullock, where she focused on the DNA binding domains of transcription factors. Most transcription factors have a DNA binding domain that allows them to recognize certain DNA sequences. But then what is typically not looked at are these large unstructured regions. They somehow recruit the transcriptional machinery by mediating interactions with some of these components. And this interaction is crucial because this allows the entire transcriptional machinery to assemble on the DNA in order to be able to initiate transcription. So these regions are what are called as transactivation domains, and these are also typically abbreviated as TADs, which is not to be confused with topologically associated domains. So what we then wanted to do was to ask, can we discover TADs in human transcription factors or transcription factors of any given organism through this IDR screen approach? And this slide basically highlights that there's no clear sequence motif that can unify all these TADs. So it's not just a simple matter of aligning the sequences correctly, because there is enormous degeneracy in these sequences that can act as TADs. So here are some published structures of TADs that can adopt an alpha helical conformation when they recognize some of the subunits of the transcriptional coactivator complexes. So the question that we asked was, can we discover transactivation domains? And if we can discover them, can we also learn what makes certain disordered sequences function as transactivation domains? And to do this, it is very important to have a very good assay. And there are many different assays, and therefore any study that we perform is going to be limited by the kind of assay that we do. So I'd like to highlight that some of the conclusions that we make are also dependent on the kind of assays that we use. So here we used an assay that involves a particular transcription factor called HSF1, which is a yeast heat shock transcription factor. So normally, when you increase the temperature, HSF1 is activated, and HSF1 binds to promoters of many different genes and initiates transcription that launches a heat shock response, and this leads to cell survival. Now, we created a version of yeast that removed the known transactivation domain. So you have, in a sense, converted an activator to a repressor. So when you increase the temperature to 37 degrees, none of the yeast cells grow because they can bind to the promoter but cannot initiate transcription. Now what we then did was to take a library of random DNA sequences that codes for intrinsically disordered regions, and then we created a library of these fusion proteins. Okay? And then we asked through the selection system which of these random members of this random library sequence can actually confer survival. So here's the list of the complexity of the sequences that we started off with. Some sequences function as an excellent TAD, so these confer survival, and they end up kind of occupying a large fraction of the population after the selection experiment. So in this manner, we looked at what sequences were tested and which sequences were functional as transactivation domains. And then we converted every sequence that was functional and every sequence that was not functional and described them into a feature vector that describes various aspects of the protein sequence, and applied various machine learning rules in order to be able to come up with some very basic patterns. And through these basic patterns, we then designed new libraries that then was subjected to a similar selection experiment in order to be able to refine the definition of what constitutes a transactivation domain. Now, in the interest of time, what I'm going to do is to just focus on our interpretation of some of the key features and the patterns that emerge as transactivation domains. So we do find an enrichment of negatively charged residues, but we also find an enrichment of aromatic residues. And what is key is that these residues tend to be closely spaced, and there are many such occurrences of what are called as these mini motifs, which are combinations of negatively charged residues with hydrophobic residues that are interspersed in between them. So the negatively charged residues ensures that the polypeptide segment does not non-specifically bind to the DNA. What we also found is that there is a requirement to have no positively charged residues in these disordered regions, otherwise it could interact with the phosphate backbone of the DNA. And the hydrophobic residues are critical to mediate interaction with the cofactor and the coactivator complexes. So this is what constitutes a TAD, and using this, we also created the most simplistic and a minimal TAD, which is a repeat of tryptophans and aspartic acid stretches. And this TAD, this particular sequence, can function as good as one of the strongest TAD, which is VP16. And by increasing the repeat length or shortening the repeat length, you can now also tune the strength of these transactivation domains. And this can have interesting applications, which I'm very happy to talk about later. So using IDEA Screen, so I just highlighted one specific part of the study. Using IDEA Screen, we studied transactivation domains. And in this study, which is just published in Molecular Systems Biology last month, we were able to discover new transactivation domains from random libraries. And for known transcription factors, we could also identify which specific region. So for some transcription factors, you have about 400 amino acids. And within this 400 amino acids is about 20 amino acids that actually function as a TAD. So which region is the one that is relevant? So we tried to apply this IDEA Screen approach to discover the functional regions that function as TAD within known transcription factors. And we also assess the impact of polymorphisms in the human population, as well as like disease mutations that have been implicated in cancer, and ask whether these mutations can have an impact on the TAD activity or not. And we also learned some of these rules and properties of sequences that could function as TAD, and we also designed these new sequences that can function of TAD with desired strength. So you can tune the strength like in a rheostat. You can have strong TADs and weak TADs, and we can also have an algorithm that now allows us to scan genomes to predict transactivation domains and the effect of mutations from looking at polymorphisms and completely sequenced genomes. All right, in the last 5 minutes or so, what I'd like to do is to take a step back and present a very short discussion about how protein disorder could increase the organismal complexity of extant organisms. So one of the big questions in biology in general is to understand what are the mechanisms that can generate organismal diversity and complexity in nature. Now, if you look at the different mechanisms that have been explained and discussed extensively in the literature, they can be broadly classified into one of these three categories. So the first mechanism basically says you evolve complexity through the creation of new components or new genes. And Sushima Ono, in as early as 1970, highlighted that gene duplication is a major mechanism by which new genes can be created, and through divergence, it could acquire new function, and therefore you could increase the complexity of the organism. Very soon after, King and Wilson basically proposed that you could also increase complexity during development by controlling which genes are expressed and which genes are turned on. And this is another mechanism by which you could now increase the complexity of the organism. And the era of systems biology, it is also very clear, numerous studies have basically shown that you could evolve and emerge complexity by creating new connections among existing components. So small alterations to which proteins can interact, like which signaling protein or which kinase now recruits what protein as a substrate during development or in a particular response, can actually have an impact on downstream phenotypes. So you could either create, or you could control, or you could connect existing components in order to increase the complexity. And this basically explains one of the age-old parable, which is that the number of proteins that are encoded in the genome is not the only determinant. When the proteins are made and how they're connected actually matters in terms of emerging and evolving complexity. So where exactly does protein disorder fit in this broader scheme? So in the link of genotype to the phenotype space through the networks of molecular interactions, single amino acid substitutions within protein coding regions in disordered regions can have a significant impact by rewiring protein interaction networks. And this could therefore lead to disruption of existing connections or formation of new connections in a very, very rapid manner during evolution. So in this manner, we feel like protein disorder can actually generate enormous diversity by rewiring networks through very small number of mutations. So even though organisms may be closely related by 1 or 2 point mutations that can either create a new substrate for a developmentally important signaling kinase, or by creating new protein-protein interactions that now allows proteins to be regulated in a slightly different way, you could very rapidly create complexity in cellular processes and thereby affect phenotype of these individuals. So what I'd like to conclude is that one could view protein disorder not just as passive linkers between protein domains, but these could be a powerful medium that can fuel evolution of organismal complexity and diversity through the creation of novel and new networks of interactions. So I'm not saying that disordered proteins is more important than structured domains, or that structured domains are more important than disordered proteins, but the structured domains and disordered regions could be viewed as the yin and yang of a polypeptide. So they have contrasting properties that allows individual proteins that have more than these structured domains and disordered domains to have much larger functional versatility. So structured domains have clearly defined biochemical properties. However, it's very difficult to regulate these defined biochemical properties. Disordered regions on their own are less functional because they don't have any defined biochemical property. However, by tagging disordered regions to structured domains, you could regulate the biochemical activity either through post-translation modifications or tuning their stability in cells. Structured domains tend to evolve much more slowly, so you have conservation aspect that is inbuilt into structured domains. However, disordered regions tend to evolve very, very rapidly, so it allows organism to use the same biochemical function that is mediated by structured domain, be it DNA binding domains or kinase activity, in very different contexts because it can bring new proteins to a structured domain that can have a particular activity of interest. So to just recollect, the kinase will still remain a kinase, but which protein gets recruited as a substrate can now be determined by these short disordered regions that can evolve very, very rapidly. So the synergy between disordered regions and structured domains therefore increases the functional versatility of proteins in organisms. And not surprisingly, many organisms in completely sequenced genomes are not purely structured or not purely disordered, but a majority of them have a combination of structured domains and disordered regions, many of which can be actually quite actively regulated by the mechanisms that I described. All right, so with this, I'd like to conclude my talk, and I'd also like to acknowledge a number of colleagues and co-workers without whom many of these work would not have been possible. I'd like to thank Maria Bullion, who was a super talented postdoc in the group, and she was a PhD student with Alex, who joined my group afterwards. I'd also like to thank Guillaume, Sebastian, Gunther, Monica, and Alex for their contribution to the project where I described the tissue-specific splicing of protein networks involving disordered regions. The work describing the gene fusion was done by another very talented PhD student, Natasha Latisheva. I'd also like to thank Maria, Rob, Tillman, Louise, Matt, and Julian for their contribution to this particular project. And the work that I described on Idea Screen was something that was spearheaded by another very creative PhD student who then ended up doing a short postdoc in my group, Charles Ravarani. Alex Erkine was the person who developed the assay that I described, the heat shock transcription factor assay. So Greet, Tamara, and Daniel contributed to various aspects of this project. And this work is just being published in MSP. So if anyone's interested, I'm happy to talk more or also refer you to this publication here. I also feel extremely fortunate to have worked with an exceptionally talented group of PhD students and postdocs in my group. And also I'm thankful to a number of funding agencies who have been very generous and kind to support several of the research activities that has been going on in my group for the last 12 years. but last but not the least, I'm also most grateful to several of my mentors who have been very generous and kind in their time and in their advice, and to also help me at various stages during my career. First and foremost, I'm really grateful to Sarah Teichmann, my PhD advisor, without whom I would not have been able to do a PhD, because she really fought for me to get a PhD fellowship, since I did not have a master's degree to apply for a PhD position there. And Aravind, from whom I really learned the importance of being able to study biological systems at each and every level of complexity, from the molecular level to the systems level, and also to understand how they evolve and how processes evolve. And Cyrus has been a real fantastic mentor and colleague who's had very important impact through his own publications and also through his mentoring before I started my group and after I started my group. I'm also very grateful to a number of mentors at various stages of my career who have given me very important advice: Venki, Veronica, Tom, and Michael, and my teachers and collaborators and colleagues for their contributions throughout various stages of my career. I'd also like to take this opportunity to advertise that we have 2 positions that are available in our group. So if any of the work that I presented here interests you, and one of the works which I didn't present, which is on the GPCRs, which I'm happy to talk about, please do look at an advert that has been posted on the job notice board. There's one position for a senior computational biologist and one position for a postdoc to work on the idea screen project that I described. That's available very soon. Thank you all for your attention, and I'll be happy to answer any questions that you may have.
Thank you, Madan, for a beautiful talk.
Thank you, Christine.
Exposition of the yin and yang and proteins. Thank you very much. Madan,
Sorry? Yes. Okay.
So a question about the IDRs that you described. You talk a lot about their role in rewiring networks, and also how in fusion proteins they can play a big role in driving cancer.
Yeah.
Do you see differences in different organisms and how carefully IDRs are controlled or used, perhaps especially in the naked mole rat, for example, that's resistant to cancer?
Yeah, it's a great question. So we haven't looked at naked mole rat in particular, but we have looked at evolution of IDRs in different genomes. So plants use a lot more IDRs, but bacteria don't use as many IDRs. And one reason why it has been speculated is that because of the absence of subcellular compartments, they become also very accessible to proteases, and these proteins can be much more rapidly degraded. And given the increase in complexity through evolution of more transcription factors, evolution of more signaling proteins, IDRs served as a perfect platform to be incorporated in other proteins that allows them to be much more tightly and exquisitely regulated. So the contribution to increased complexity could come from exquisite regulation that may not be obvious from looking at purely the structure domain content of proteins.
Thank you.
Yeah.
Madan, have you looked at the non-IDR regions that are at the cusp of turning into by means of a few number of mutations in the future?
Yeah, that's a great question. So we have been looking at an aspect which is still an ongoing project that's unpublished, where we have looked at these what I call as the twilight zone, where you look at the hydrophobic residue composition. There are many of these proteins that don't have sufficient hydrophobicity to fold, and those that do have. So Keith Dunker had presented some unpublished results where he showed that in the case of zinc finger proteins, you have evidences where mutations allow certain regions right around the zinc finger region to acquire a defined fold or very rapidly be lost. And we think this could have an impact on the ability to dimerize or act as monomeric proteins. So you could tune the oligomeric state of proteins by having these IDRs that during evolution among orthologous proteins, for instance, could acquire slight structural context or lose structural context, and that can actually contribute towards this oligomeric state dependent regulation of protein function. Yeah.
Hi, thanks for the nice talk. I want to introduce this by saying I'm not a molecular biologist, but I am a bio-ontologist. So especially during the splicing portion of your talk, but also elsewhere, I wondered if we're heading into a naming problem. So how do we know, you described some proteins that had almost opposite effect on pathways or function downstream. So how do we know that we're using sort of the right name for a protein at the sequence level when it has kind of opposite functions depending on the splicing or these?
It's a very important question because I think in the field, particularly when molecular biologists work on single proteins, they refer to a unique name, so they call it by the isoform. But when you are working at the level of entire systems or entire networks, you could use a PetitNet framework where you can have different states, for instance, that could be encoded. But beyond that, it's very difficult to, it is an ontology problem that needs to be brought in. And that's why referring to proteins and genes or protein products and transcripts through their molecular composition is the only way to say that there is a distinct chemical entity that is different, that comes from the same gene product. So I don't have an answer to this, but it is an ontology problem that could be looked at more seriously and closely.
Great, thanks.
Yeah.
Hi. Thank you so much for that talk. It was truly inspiring and fantastic. I had a question about the mechanism of how this occurs. Could you tell us a little bit about the entropy of binding of these highly disordered regions, please?
That's a really important question, so I didn't get into this in the interest of time. So when you have a biomolecular interaction, the fact that they're extremely flexible and when they undergo what's called coupled folding and binding, there's an entropic cost. So they undergo from a disordered state to a fully ordered state, and there's an entropic cost. But then there's also an enthalpic gain, and the enthalpic gain comes from the association of this folded state with a structured domain. But then when you look at the entropic cost and the enthalpic gain, they're comparable. So you have a system where you have an entropy enthalpy compensation, and therefore you can have highly specific association but not very strong affinity. And this is excellent for certain processes like signaling, where you want interactions to be specific but not a high affinity event, because a single post-translation modification can now affect the enthalpy sufficiently to be able to disrupt this balance, and thus the protein can dissociate very quickly. So it's a very good example of a thermodynamic system/state of protein-protein interaction where you don't need to have large surfaces to have highly specific high affinity interactions, but you could still have highly specific low affinity interactions mediated by these disordered regions. And many of these are in the cusp segments where they have some hydrophobic residue but not sufficient to autonomously adopt a fold. But when the right chemical surface gets presented, they actually now adopt a fold. Yeah.
Hi, I noticed that your phylogenetic tree was dominated by macroorganisms, and I was wondering if you could comment, splicing aside, to what extent these mechanisms you discussed apply to prokaryotes.
So to prokaryotes, there are 2 or 3 major places where disordered regions feature quite prominently. One is in regulating the half-life of proteins. So there are tags that in certain prokaryotic organisms called pupillation that gets added onto proteins that are degraded by the CLIP-PX system. So there's one very clear example where certain disordered segments have been used as tags for tuning the half-life of proteins. And the other major place where disordered regions have been used in prokaryotes are in membrane proteins, where the cytosolic side of membrane proteins are conformationally flexible and also heavily modified, that allows recruitment of, let's say, the transcription factor, like in the case of the histidine kinase sensor system. Disordered regions are featuring more prominently. And then the third place where disorder also contributes in the case of prokaryotes involves the single-pass transmembrane helices that can actually act as a repressor for the histidine kinase system. So you have these single-pass TM regions that get inserted, and then you have these large flexible tails that are inserted onto the membrane that can actually block the histidine kinase domains on these systems. So these are some examples that I could think of where disordered regions have been used in prokaryotic system.
Over here,
yeah.
Other side. Very interesting talk, thank you. I'm wondering about what you said concerning alternative splicing of disordered proteins, where you pointed out that a main effect is to rewire the interaction network. And I'm wondering, is that specific to disordered proteins, or does the same happen also for structured proteins? And if not, what is the different effect of alternative splicing?
That's a great question. So I think Christine has some work where she's also looked at mutually exclusive exons, so where there are structured domains that are included or excluded in a mutually exclusive manner that have single point mutations that allow rewiring of interfaces. So that is one very well-known example of structured domains being alternatively spliced. What I want to also highlight these are only tissue-specifically spliced exons. So I don't want to say that all alternative splicing only affects disordered region, but the tissue-specifically spliced regions are enriched in disordered processes. And beyond biomolecular interactions, you can also have autoinhibition as a way to modulate binding, and also you can have conformational entropy that could tune the affinity without affecting specificity. So these are the other processes that could be mediated by disordered regions. Yeah.
I think we need to stop there because we've overrun, but I just want to thank Madan again for a very beautiful, very inspiring talk, and to award him his prize. Yes.
Thank you, Kristen. Thank you.